All right, so my research was on this bacteria. It's called Pseudomonas aeruginosa, and it's a really important pathogen in clinical settings because it's an opportunistic infection, which means that if you have something that is affecting your immune system or making you more tired or just generally taxing your system, this bacteria is going to take advantage of that, and it's going to go like hog wild. And so you see this a lot in cystic fibrosis patients, which is, they already have a lot of problems with breathing and infections already because of how their mucus system works. And so this infection just really takes hold there. If you have catheters or other implants, it takes hold in those as well. And it's really, really dangerous in burn victims because you have to keep burn victims, their wounds moist. And of course, bacteria love warm, moist things. And so it's really just important. But we also see this a lot in otherwise healthy patients because this bacteria is really virulent. And so if you've ever had swimmer's ear or anything like that after you've been swimming in a lake, it's most often going to be this Pseudomonas aeruginosa. And so this is kind of cool because we have a huge population of Pseudomonas aeruginosa in Germania Springs, which is right down the road. And so it's really easy and cheap to do research on it. And so that is the main reason why this specific bacteria was chosen. And so the point of this research was to see how the Pseudomonas aeruginosa populations were kept in check by other bacteria in the community. And so Pseudomonas aeruginosa and a lot of other bacteria, they create these things called biofilms, which are it's like a mucus-type substance that they secrete to protect the population as a whole. And so what happens is you have all these cells in the middle and this thing called biofilm around it. And that makes it so that if you have antimicrobial substances, like antibiotics, anything like that that's trying to attack and kill the cells, it has to get through the biofilm first. And so it's really hard to kill them. But what we've seen in a lot of research is that Pseudomonas aeruginosa is one of the first bacterial species to set up these biofilms in water and aquatic systems. But they're also one of the first to get kicked out by other bacteria. And so we were like, well, how is that happening? Why? They're so good at taking hold in human bodies and other, in mammal and causing infections, so why are they so easily kicked out? And so the Pseudomonas aeruginosa biofilm structure is different from most biofilm structures because it relies really heavily on extracellular DNA to keep its structure. And so this mechanism that I proposed was that the other bacteria in this biofilm were producing this thing called DNase A, which is a type of enzyme that breaks down this DNA, and that's allowing the bacteria to break down the biofilm, the protective layer, and get to the cells and kill them. And yeah, so that was the research. And so the two bacteria that I tested were I just went out and got a whole bunch of water samples and swabbed and swabbed and swabbed and swabbed until I found two that were proven to produce DNase A. And so then I went through and I tested them with, it's called Pseudomonas aeruginosa extract, which is a fun way of saying that I went and I cooked up a whole bunch of Pseudomonas aeruginosa, and then I took the cells out, so it was just the biofilm. And so there's no cross-competition with growing on your Petri dish, it's just the biofilm. And so I put that in the DNA, in the media that I grew them on. And then I tested how much DNase A they produced. And kind of excitingly, I found that the presence of this Pseudomonas aeruginosa extract does cause an upregulation in this DNase A production, so that there is at least one of these bacteria is producing more DNase A in the presence of P. aeruginosa. And this was a really simple experiment, first off, to just go, is this actually happening, before we put a lot of time and money into it. And so for this next phase that I have, since I have proven that something is causing an upregulation, we're going to figure out what it was. And so I had 2 environmental isolates out of about 30 that produced this DNase A. One of them was, it was proven statistically that it had an upregulation, and one of them didn't. And so my assumption on this, and what I'll be testing this semester, is that one of these bacteria, whether it's the one that didn't or did produce the upregulation, is just a different strain of Pseudomonas aeruginosa. And it's breaking down its own biofilm to use it for something else, because that's been proven to happen. And the other one is another bacteria. And so this semester, I'm going to do this. I'm going to create the media again. It's going to be much more refined this time, and I have a budget this time, so it'll be good. And it's going to be a much more refined process of purifying this extract to make sure that we have, and quantifying the actual quantity of DNA in this, and then testing it again with lab cultures. So we're going to buy several bacteria from some fancy lab, so we know exactly what it is, and so I don't have to go out and find bacteria, and then test it. I'm going to test 3 bacteria that produce biofilms, and then as a control, I'm also going to test a Pseudomonas aeruginosa strain as well to see if it's the Pseudomonas aeruginosa producing the DNase A to kill itself as a competition. Cool. So yeah, so that's what I'm going to be doing. It's exciting to me, at least. I'm enjoying it, and it's a lot. And it was fun to do. So it's fun to learn how to do something on a 0 budget. Yeah. So, do I have any questions?
You have a future in higher ed.
Yeah.
On a 0.
I have just a quick question on this.
Okay.
So people usually think if it's a spring, they can drink out of it. And it's
No.
water.
Yeah.
You're saying we should not be drinking Jiminy Springs?
No, definitely not. And for the most part, as long as you do that thing, where you get out of the lake and you shower off, it's not going to cause any problems unless you're already immune compromised. And if you know that already, for the most part, so you'll be fine. If you accidentally drink some water, it's not going to be a problem. This is just if you're negligent about it and don't keep proper hygiene, then yeah, it's going to be a problem. But for the most part, no, not really.
I'm just curious, how did you come to find out that Jiminy Springs is so loaded with stuff? I mean, that's kind of creepy.
It's just because my concentration in biology is microbiology, and it's really fun to just swab stuff and see what's growing there. And so this is just a common thing that we do. And then we found it when I was taking microbiology last spring. We found a whole bunch of it, and so we were like, might as well continue. So then we did that. Yeah.
Are there any other places we should be aware of?
Maybe the potting soil.
Right, yeah. Stay away from that.
That would be great.
That was
Well
cool.
done.
That's neat from an ecological perspective too. And there's a lot of
Yeah.
really interesting interactions going on that we don't normally get to study very easily from aquatic systems, particularly because biofilms are so critical for our systems.
Oh yeah. Yeah, because the biofilm, anytime you step in a lake and if the rocks are kind of slick, that's the biofilm that we're talking about. So it's really cool.
Great.
Yeah.
That's what my snails are eating. That's
And
what my
don't
snails
look
are
there
eating.
either.
Yeah. So don't lick their snails.
Don't look there.
Eric, let's thank our speaker again.
And thank you to Dr. Case for